Chapter 16, Part 3. So, we have a couple of things to talk about with this here. We've got major histocompatibility complex. That is also called MHC, because it's a mouthful. And then those are proteins. Major histocompatibility complex proteins are on the surface of antigen-presenting cells. So the MHC are proteins, or antigens, that we first identified in graft or transplant patients. So it turns out that now these are really important in determining compatibility for tissue transplantation. So when they are checking to see is someone compatible with another person, can they take a kidney or a liver or whatever and put it in this person, they're looking at MHC, and they're trying to find the versions of the proteins that are similar enough to convince the recipient's immune system that these tissues are normal, that they belong there. So these proteins are glycoproteins. These MHC antigens are glycoproteins. They're in the membranes of most cells. This is not a human thing. It is a human thing, but it's a reptile thing. It's a bird thing. It's a fish thing. Basically, almost all cells of almost all vertebrates have these kinds of proteins, these MHC kinds of proteins. Of course, each species has its own specific versions of them, but everybody has them. Who's a vertebrate, anyway. And what these proteins actually do is they have antigenic epitopes, and they present it to immune cells, to cells of immunity, and say, this is what I have, or this is what I am making, or this is what I just did phagocytosis on. And they display it and they show it to the immune cells, the lymphocytes. So these antigens bind in what's called the antigen binding groove of the MHC molecules. There are two basic types of MHC proteins. Class 1 is on every cell in your body except the red blood cells. And class 2 is only present on a few cells that are actually called antigen-presenting cells. These are macrophages and dendritic cells. So these guys these guys will do, for example, they'll phagocytose something, break it into pieces, take some of those pieces and put them into the MHC proteins that macrophage is making right now. And then that all gets transported to the plasma membrane, and then these immune cells will come along and check and see what the macrophage just ate, or what this virus-infected cell is producing right now. And this is the structure of them. Class 1 MHC, this is again on every cell except red blood cells. Every cell that has a nucleus, which is everything but blood cells. At this end, there is this antigen binding site, this groove. And this is where the cell will put antigens, the things that they're making. They're just saying, hey, this is what I'm making today. And they just give them a little sample of what they're up to inside this little cells factory. The MHC or B cells, the class 2, or on other antigen-presenting cells, again, we have a groove. This time we have two peptides, both of which penetrate the plasma membrane. On that class 1, only one of the proteins goes into the plasma membrane. These are two separate proteins. Actually, these are four separate proteins. They're held together by disulfide bonds. Here, and same with here. So these are class 2. These are only on certain kinds of cells, antigen-presenting cells, or B cells. So cells do what's called antigen processing. So depending on what kind of cell it is, they're going to just take random piece of protein that they just made and process it to add it into those MHC proteins for display. Now there's different processes, whether it's an endogenous product or an exogenous product, right? Is this something that this cell is making, or is this something that this macrophage just did phagocytosis on? So we'll have to process those differently. So for endogenous antigens, the MHC proteins get produced by ribosomes that attach to the ER, and so this is how they're oriented inside the endoplasmic reticulum. And meanwhile, randomly, proteins are just plucked out of the cytoplasm, broken down into epitopes, and transported into the lumen of the rough ER, where they associate with these class 1 MHC proteins. These get packaged into a little vesicle, they bud off, they move to the Golgi, and they will eventually end up on the plasma membrane. And here is our class 1 MHCs on the surface of the cell, displaying each of them a different epitope, a different piece of something the cell was making. Now, if this is an endogenous antigen, and that means that it was made by this cell. That doesn't mean that it was genetically a part of this cell. In other words, this is a way that your immune system checks every cell for, are you virus infected? If this cell has a virus infection, at some point one of the virus proteins are going to get picked and get degraded and get presented on these MHC proteins. And then when this cell gets checked out by the T cells, it will trigger a response because it's making things that doesn't belong. So this is important, not because most of the time, most of what your cells are making is totally normal. But when there's a virus infecting them, this is a really nice way to be able to scan them and find the ones that are making things they should not be making. In this case, we have a cell that's doing phagocytosis, so it ingests and digests the particles, and so we have a phagolysosome. So we've digested that bacterium or whatever it was, and now we are going to see fusion with the class 2 MHC proteins embedded in the membrane of this vesicle. So these two are going to merge and fuse, and then these little epitopes are going to associate with these class 2 MHC proteins. And then they are going to get transported out to the surface, and again, this makes them available for immune cells to come and say, find out what is it that this cell just did phagocytosis on. And is there more of that, and what do we need to do about it? So in the next section, we're going to talk about the T lymphocytes and what happens to cells when the T lymphocytes find that they have either digested something interesting or that they are making something that they should not be making.